Bismillah ar-Rahman ar-Rahim. Our lecture is about lipid transport. Fat absorbed from diet and lipids synthesized by liver and adipose tissue must be transported between various tissues and organs for utilization and storage. The problem arises of how to transport nonpolar lipids, which is the triacylglycerol and cholesterol esters, in the aqueous media, which is the plasma. The problem is solved by incorporating nonpolar lipids with amphipathic lipids, which is the phospholipids and cholesterol, and proteins to make water-miscible lipoproteins. So, what are these lipoproteins? Plasma lipoproteins are spherical macromolecular complexes that are composed of lipids and proteins. Its function is to allow lipids to move through plasma inside and outside cells, as you can see in the figure. This figure shows us the structure of lipoprotein, which is composed of hydrophobic core of triacylglycerol and cholesterol esters. This core is surrounded by a single layer of amphipathic apolipoproteins, phospholipids, and non-esterified cholesterol. The amphipathic components of the surface layer are oriented so that their polar groups face outward to the aqueous medium and their nonpolar groups face inward. Note that in lipoproteins we have one surrounding layer of lipoproteins, not two like in case of cell membranes, because the inner environment here is hydrophobic, unlike the inner environment of the cells, which contains water. Remember that phospholipids are molecules of fat attached to a phosphorus containing a group, while amphipathic compounds are compounds with polar and nonpolar parts. We have several types of lipoproteins that differ in the ratio of lipid to protein. Also, they are different in size and density. Lipoproteins are classified based on their density, ranging from high-density lipoproteins, HDLs, low-density lipoproteins, LDLs, intermediate-density lipoproteins, IDLs, very low-density lipoproteins, VLDLs, and chylomicrons. Lipoprotein size is inversely related to their density. For example, HDL particles are the smallest in size and the densest of the lipoproteins, as you can see in the figure. Chylomicrons, on the other hand, are the lipoprotein particles lowest in density and largest in size. Note that chylomicrons compose of about 99% of lipid and 1% of protein. Of the 5 types of lipoproteins, chylomicrons contain the highest percentage of lipids and the lowest percentage of a protein. So, as the lipid to protein ratio increases, the size of lipoprotein increases and the density decreases. Because protein density is larger than lipid density. اوكي هلا كملخص للي حكيناه, as you can see in the slide, chylomicrons are the largest in size, lowest in density, and highest من ناحية lipid to protein ratio, while HDLs are the smallest in size, highest in density, and lowest in protein to lipid ratio. Lipoproteins can also be classified based on their mobility during electrophoresis on the agarose gel as alpha, pre-beta, and beta lipoproteins, which correspond to HDL, very low density lipoprotein, and LDL respectively. HDL is the smallest in size, move faster as you can see in the figure, so high density, smaller weight, longer distance. Apolipoproteins are the protein component of lipoproteins associated with lipoprotein particles on the outer surface, as you can see in the figure. These lipoproteins have several functions. For example, they provide recognition site for cell surface receptors, also serving as activator for enzymes involved in lipoprotein metabolism. We have 5 major classes of apolipoproteins, A, B, C, D, and E, and these classes are then subdivided into subclasses, for example, ApoB48. As you can see in the figure, the most prominent apolipoproteins For example, ApoA found in HDL, ApoB in LDLs, chylomicrons, IDLs, and VLDLs, and ApoC in VLDLs and IDLs. Okay, now we will start talking about each lipoprotein in detail, starting with chylomicrons, which are lipoproteins responsible for transport of lipids from intestine to the circulation. Apolipoprotein B48 is the apolipoprotein found in chylomicrons. Its synthesis occurs in the rough endoplasmic reticulum of the small intestine as a result of RNA editing process. Okay, هلا شو اللي بيصير عندنا؟ In liver, the single ApoB gene is transcribed into a messenger RNA that directs the synthesis of a 100 kilodaltons protein ApoB100. إذا اسمه البروتين ApoB100 وهذا ينتج من الليفر. In the intestine, the same gene directs the synthesis of the primary transcript. However, a cytidine deaminase converts a CAA codon in the messenger RNA to UAA at single specific site. So, rather than encoding glutamine, this codon becomes a termination signal and a 48 kilodaltons protein اللي هو ApoB48 is the result. ومن هون إجت تسمية ApoB48. ApoB100 and ApoB48 have different functions in two organs. So again, two organs اللي هم الintestine which produce ال ApoB48 and liver which produce ال ApoB100. Synthesis of triacylglycerols, phospholipids and cholesterol occur in the smooth endoplasmic reticulum. As I said earlier, synthesis of ApoB48 occurs in the rough endoplasmic reticulum. So, after their synthesis, both ApoB48 and the lipids from SER incorporated to form chylomicron. This formation occurs during the transition from ER to Golgi, where particles are packaged in secretory vessels. These vessels fuse with the plasma membrane, releasing chylomicrons, which then enter the lymphatic system and ultimately blood. The released chylomicrons are called nascent chylomicrons because they are functionally incomplete. When it reaches the plasma, the particle is rapidly modified, receiving ApoE and ApoC, which is necessary for the activation of lipoprotein lipase. Lipoprotein lipase enzyme degrades the triacylglycerol contained in the chylomicron. The source of these apolipoproteins is circulating HDL. So, chylomicrons received apolipoprotein E and apolipoprotein C from circulating HDL. This table shows us the percent composition of lipid fraction. Note that chylomicrons contain the largest percentage of triacylglycerol with about 90%. Also note that LDLs have the highest percentage of esterified cholesterol with about 35 to 40%. Reaction with lipoprotein lipase result in the loss of approximately 90% of triacylglycerol of chylomicrons and in the loss of ApoC, which returns to HDL but not ApoE, which is retained. The resulting compound, called chylomicron remnant, is about half the diameter of the parent chylomicron and is relatively enriched in cholesterol and cholesterol esters because of the loss of triacylglycerol. Chylomicron remnants are taken up by the liver by receptor-mediated endocytosis, and the cholesterol esters and the triacylglycerols are hydrolyzed and metabolized. Uptake is mediated by ApoE. Triacylglycerols hydrolyzed by lipoprotein lipase give fatty acids and glycerol. Fatty acids are stored by the adipose or used for energy by the muscle, while glycerol is used by liver, for example, in lipid synthesis, glycolysis, or gluconeogenesis. Lipoprotein lipase synthesis is stimulated by insulin. Move to the second type of lipoproteins, which is the very low density lipoprotein produced in liver, carry triacylglycerol from it to peripheral tissue where it is degraded by lipoprotein lipase. Very low density lipoproteins are secreted directly into blood by the liver as nascent VLDL particles containing apolipoprotein B100. They must obtain ApoC2 and ApoE from circulating HDL. As I said earlier, ApoC2 apolipoprotein is important for the activation of the enzyme lipoprotein lipase, which degrades triacylglycerol to fatty acid and glycerol. Reaction with lipoprotein lipase results in the loss of approximately 90% of triacylglycerol of VLDLs and the loss of ApoC, which returns to HDL but not ApoE, which is retained. The remaining triacylglycerols are transferred from VLDL to HDL in an exchange reaction that at the same time transfers cholesterol esters from HDL to VLDL. This exchange is accomplished by cholesterol ester transfer protein, as you can see in the figure. These changes occurring to VLDL lead to the formation of VLDL remnants or IDL, اللي هي intermediate density lipoprotein. After metabolism to IDL, these IDL could be either taken by liver via receptor-mediated endocytosis that use ApoE or ApoP100 as the ligand, or converted to low-density lipoprotein LDL. Low-density lipoproteins, LDL, are the lipoproteins with highest percentage of cholesterol esters. Their function is to provide cholesterol to the peripheral tissues. They do so by binding to a negatively charged glycoprotein receptors that recognize the apolipoprotein B100. These receptors are clustered in pits on the cell membrane. The intracellular side of the pit is coated with protein glycerine, which stabilizes the pit. After binding, the LDL receptor complex is internalized by endocytosis. The vesicle containing the LDL rapidly loses its glycerine coat and fuses with other similar vesicles, forming large vesicles called endosomes. The receptor then migrates to one side of the endosome, whereas the LDLs stay free within the lumen of the vesicle. The receptor can be recycled, whereas the lipoprotein remnants in the vesicle are transferred to lysosomes and degraded by lysosomal enzymes to give amino acids, fatty acids, and cholesterol. Elevated levels of endocytosed cholesterol reduce the activity of HMG-CoA reductase enzyme, so reduce cholesterol de novo synthesis. It also reduces the expression of LDL receptor gene, so limiting the entry of LDL cholesterol to the cell. High-density lipoprotein, or HDL, is considered as the good cholesterol. High levels of HDL are better for our health because they remove other forms of cholesterol from the bloodstream. HDL is synthesized and secreted from both liver and intestine. HDL particles serve as circulating reservoir of ApoC2 and ApoE. However, ApoC and ApoE are synthesized in the liver and transferred from liver HDL to intestinal HDL when the latter enters the plasma. We have an enzyme called phosphatidylcholine cholesterol acyltransferase. This enzyme binds to nascent HDLs and is activated by ApoA1. شو بيعمل الإنزيم؟ It converts phospholipids and free cholesterol into cholesterol esters and lysophosphatidyl-choline. So this enzyme converts الكوليسترول free cholesterol اللي جاي من cell membranes ومن ال other lipoproteins. It converts it into cholesterol esters and converts الphospholipids into lysophosphatidyl-choline. الكوليسترول esters شو بيصير لها؟ It move into hydrophobic interior of the bilayer, whereas the lysophosphatidyl-choline binds to albumin. So بيصير في عندنا non-polar core forming a spherical pseudomycellar HDL covered by a surface film of polar lipids and apolipoproteins. This aids the removal of excess unesterified cholesterol from lipoproteins and tissues. ومن هون بتيجي أهمية ال HDL lipoproteins as a good cholesterol.